Okay, let's start off the course today. If you can hear me always, we check the voice and audio is working. So, I mean, if you can hear me, please raise the hand. Okay, so then I can see. Okay, so it's working. So we are on the session 3 and 2 on mutation classification, right? And I have little announcement for the your first assignment on next Monday. So as I introduced in the first session, we have 2 assignments in this semester. So the assignment is basically ask you whether you understand the sort of the basic concept you learned over the topics, and 2 assignments instruction is included in the first lecture slides you find in the GitHub link on here. And if you go to the introduction slides, then you can find the link to the, what you have to do in your assignment. And there's all introduction for your assignment. This is the first one, right? And you have the second one. And the first one is the due date on, so you have to check the due date. Okay, to here. So if you go to our Blackboard, you go to the tab called Assignment and Test, then you will find the link for the first assignment, the submission page. So if you click this, then you can identify the due date on that. So it's a Monday, 3rd, 11:59 p.m. So if you have read on this page, please prepare your first assignment and submit on time, right? So I think I put it on the instruction, and it has to be on time submission. So late submission is not allowed. So and also the first assignment is based on the scientific publication. So some of you might not really familiar with reading the stuff. So what I can tell you is that prepare in advance, right? So everything you have to schedule your time and check up the, when is the submission date and how you schedule your time with other, subject studies and, midterm exam preparation. So be on that preparation and also schedule your time. This is the one-on-one things for your career. So I experienced some of the students, they excuse that your submission is late and there's any second chance, but we have the 2 months in advance announcements for the assignment. So what else I can do that, right? So this is like 12th April. It's about a month before the submission, right? So please prepare your stuff before the submission date and submit on time. So that's why I instruct way ahead on the date. Okay, so this is the first assignment, and this is basically checking the whether you understand sort of the concept you learned in the session 2 and 3, and you have to mix up your languages and also sentences to describe what you have read from the scientific paper. And every year I choose the one of the best scientific papers over the year. And this year I picked up 2. One is the Kaplanis et al. 2012 from the Nature, and this is the largest the genetic screening for the developmental disorders, right? So this is one of the critical paper. Another paper is from Science in 2018, written by me, and this is another landmark paper for the non-coding discovery. So the 2 things you can read by yourself during the time. Okay, so back to the today's topic. So we've done we cover some of the introductory part of the mutation classification last lecture, and we deal with the different ways to classify the types of the mutation from our genome, right? So we thought about the size of the variants last time, and we covered the frequency of the variant and consequence of the variant and transmission of the variant this time. Okay, then we briefly deal with the other variant types called SNVs and indel, and we are middle of the touching the topic structural variation. Okay, so what is the structural variation? This is the large-scale genomic changes, and normally described as genetic variants greater than 1,000 base pair. And we have two types of structural variants. So first one is the copy number altering variant, which is literally change the status of copy numbers in our genome. And the other one is the genetic changes without the copy number status changes, right? So copy number altering variant, we shortly call copy number variant or CNV. And this includes two types. So first one is deletion, and second is the duplication. And copy number neutral variant is the one not changing any copy number status, and it included the inversion, insertion, and translocation type. And we have more complex types of the structural variation, right? So this is the combination of more than two types of structural variants, right? So the one we called it in the here is, I mean, we can say canonical structural variation. So canonical is one of the vocabulary I felt quite confused when I was students, because there's a rare way we talked about the canonical in Korean word. So canonical means that it's like a typical, right? So its meaning is like a typical. So canonical something means that we typically see in the biological phenomena, right? So here we call the canonical structural variation, which is the, normally we call, structural variation like, deletion, duplication, inversion, insertion, translocation. This is like a textbook style structural variation. But in more recent days, we see more like, non-canonical structural variation because of the technical development, right? As the technical, as the genomic technology developed more and more, we see more exceptions. We see more in-depth genomic changes from our DNA, right? So that is, we call the non-canonical structural variation, right? So this is another way we describe the situation. And we put this situation as complex structural variation. So in that case, we have more than two types of structural variation involved in our DNA changes, right? Okay, so what we have, which types of the structural variation we have in our genome. And we need something, we have to look at more like a, global view of structural variation in our genome. So in that case, what people normally do is they create some reference data sets. So here I introduced the NOMAD project, which is the GNOMAD project. And the G is the silent sound, so we call it NOMAD project. It's like the people hang around the world. So in the NOMAD project, we can see the frequency of certain variants in our genome. So it includes the SNVs, it includes the indels, but it also provides you the information about the structural variants, right? So basically, we can ask that, someone can ask you, how many deletions or how many duplications or how many structural variations we have in our genome? How many of them can happen in one individual, right? So simple answer is that we can look at the reference data set, that we can estimate sort of the average numbers of them in our genome, right? So if you look at the NOMAD project, and now we know that there are 7,400 structural variation per individual. So we can say that, around, 70 to 80 structural variation we can find in one person's genome, right? And then I mean, what NOMAD actually did was that they look at the sort of the global profiles of the structure variation from the many individuals. So this case, they look at 14,000 genomes from the lots of people in the world, right? So I mean, we can say diverse global population. And of course, there are some people from Europe, some people from Africa, some people from East Asia or South Asia or something, right? So what you have to keep in mind that when you look at the biological studies, there are some ways to describe the reference, right? Reference here is also quite general purpose, general vocabulary. So you probably remember the reference genomic sequence, right? So that case, we have the sequence information as a reference, right? And here is reference means that here's reference meaning the frequency of certain genotype, frequency of certain alleles, right? So here has the different ways to provide the meaning of the reference, right? So reference sequence is sort of the building the sequence information from few individuals. But here we have the reference information for the genotype information. So here has the totally different aims to get the allele frequency information, right? Okay, and that is one thing. And the other one is we have about 7,000 to 8,000 structure variation per individual. And what you have to notice is that this is way less number than small variance. So small variance means that SNVs were in there, right? So you can probably ask why, right? So why is the critical things? Because so let's imagine the situation if you have the larger genomic changes in your DNA, then you can intuitively think that these large changes probably give you do something. So if there's a great chance of doing something, then the small size variant, right? So in other words, I mean, you have the gene, right? And the gene has like 18,000 base pair, right? And you have the mutation, and that mutation is the SNV. So you change one base pair out of 8,000 base pair, and that is the one mutation, right? And the other mutation you have in other person is it's like an 80,000 base pair deletion. So totally that gene is totally gone, right? So that gene is one copy is totally deleted, right? So if you compare these two mutations, so one is SNV and the other one is deletion, then you can intuitively think that this deletion must have larger impact than SNVs because it's removed one gene completely, right? So that case, you can expect the larger genomic changes probably have a larger impact on the phenotype, right? So in other words, in the population view, we can say that the larger variants probably have the larger impact on the natural selection, right? So that's why if you look at the SNVs or index or structure variation, and the numbers of the occurrence per individual is differ by the type because the larger one has to be rare in an individual, right? Okay, so that was the one point, and also the another the properties of the structure variation is that structure variation also can represent population structure as SNVs or index, right? So if you put that structure variation into the PCA plot, which is the principal component analysis plot, then you can clearly see the differences between the populations. So African population, American population, Asian population, and European population, they totally distinguished by the information of the structure variation, and we're going to learn how we distinguish them in the mathematically in a later section. And another point is that if you look at the size of the structure variation, it's ranged from the very short one to very large one. So the shortest one is like around 1,000 base pair, and the largest one is greater than 1 million base pair, which is the huge genomic change, right? And if you look at the number of singleton variant, which is the only appeared in a single individual. So singleton means that the variant happening only one individual in that data set. So that means that you look at the 10,000 individuals in here, but a certain number of variants only happen in one person in the data set. And if you look at the proportion of the singletons across the size, you can see that the largest one has the highest singleton proportion in the data set. The reason is the same as I explained in the natural selection. The reason is that the large one probably have the larger impact on the trait. So it's unlikely to be transmitted to the next generation because of the natural selection, right? So rare means that, rare variant means that it's under strong natural selection. So it's only seen in the only few individuals in a population. So you have to understand the relationship between the number of individuals and the frequency and also the natural selection, right? And also in other words, we can say that the size of the structural variation is correlated with the effect size of the structural variation. Okay, so structural variation is the important key to many disorders. So structural variation is traditionally and long been described in the medical genetics, right? So here I include one example from the autism spectrum disorder, which I'm working with. So structural variation could bring a large impact on disease, right? In a particular patient who has severe phenotype because of the large size. And structural variation, that's why a primary interest of the genetic screening in clinical genetics. So if you go to clinic during your pregnancy or the time, you are subject to the genetic screening test, and the hospital asks you whether you want to get some genetic testing, and it's covered by insurance, and that's basically testing whether you have very severe or very deleterious structural variation that can happen in the developmental disorders or congenital disorders. In that case, you are thinking about the stop or for the treatment or something, right? And also over the many years we did some studies, and we have the deep knowledge of which loci frequently happen that structural variation associate with certain disease, right? So in other words, the genomic location where the structural variation are observed in patients with critical disorders, right? So that means that the frequent means that recurrent loci, and disease associate means that the structural variation doing something on a disease, right? So we, over the many years, in autism, schizophrenia, intellectual disability, type 2 diabetes, or social disorders, people looking at those recurrent loci for the disease associated with structural variation. So we have sort of the stacks of the information for that genomic patterns. Okay, so in autism spectrum disorder, there are about 5% of the autism cases. They are explained by structural variant, and it's well discussed in 2015 studies. And in schizophrenia, 1.4% of the cases affected by structural variation, right? And this is quite interesting topic, and people still studying why that happens in our genome. And also, I'm not covering in this slide, but some of the structural variation gives you diverse phenotypic effects. So let's say here we have the 16p11.2 duplication. This is observed in the patients diagnosed for autism, but also that mutation can occur schizophrenia, that structural variation can occur the intellectual disabilities or other genomic symptoms. So diversing phenotypes by the same genomic location is frequently also observed in the human genetics, and this is one of the topics lots of people want to research in these days. Okay, and the next way we can describe the genetic variance is the frequency, right? So as I mentioned, the frequency is related to the natural selection, right? And the point is that how we can explain this natural selection explained by the genetic variant, and also how we define this, how we define the threshold, that frequency we are using in the study, right? Okay, so selection and frequency. So natural selection works on the traits, and so the frequency of the variants contribute to traits can be changed, right? So for example, if you have the variants causing death before 4 years old, which is, if you have the mutation in your genome, then you are unlikely to survive before that days, right? So in other words, that means you're unlikely to transmit your variants to the next generation. So the variant should be removed in a population, and the frequency of the variation has to be 0, right? Because there is no chance to transmit that variance to the next generation, right? So it has to be removed totally in our population. So the level of the natural selection is we can think about the how much that the genetic variant or alleles can be transmitted to the next generation. So this is linked to the probability of the genetic variant. And of course, that has to be varied by traits, and this is because all the traits are not deleterious or not lethal in an organism, and as well as disease too. So the traits and disease has varying levels of natural selection in our population. So one way that some traits are favored by selection, then in that case we can expect that increasing the frequency of the variant, right? And also, given the polygenic model, single variance is likely contributing partially, right, or highly partially to a trait. This is the way we learned in the session 2, right? So that means that there is a wide range of the frequency of variants, right? Because if the trait is determined by Mendelian factor as Mendelian model, then we have only few class of the variant frequency in our population. But as Darwin described in his model earlier, we have the wide range and we have the continuous range of the trait values, and that is actually recapitulated by the range of the allele frequency as well. So the frequency of the variants also quite continuous as the trait value. This is because the genetic variant is partially contributing to the traits. In other words, most of the portion of the genetic components is additive, right? So that's why the single variance is unlikely to be in the natural selection situation, and that's why it segregate to the next generation. Okay, so in this sense, we can think about, okay, so how we can define rare or common variants based on the frequency, right? So if you want to look at some trait and some, if you want to measure the effect of certain variants, then you want to set up the cutoff for the the variants, and then you want to define rare variant or common variant, right? And if you look at some genetic studies from the scientific journals, then they are commonly use the terms like a rare variant or common variant, and this is one of the notion that which part of the genetic studies you want to do in your future, right? So the way you approach the rare variance or the way you approach the covariance is quite different, and also the commercial application and also clinical diagnosis is also different by the rare variance and covariance. So if you look at the recent literatures on that, then you have, the terms like, describing the rare situation or the common situation. But one tricky thing is that the cutoff to define this rare variance or common variance is quite different by studies. So when the genetic studies done earlier, like 2000 or 2005, people define covariance as like a 5%. So if the allele frequency greater than 5%, then you define, okay, so this variant probably common variant, right? And less than 5% allele frequency, then you define, okay, so this is the rare variant, right? At the time, we have only like, few people we can use in the study. So that's why defining the rare and common is limited because of this number of samples, right? So 5% was the reasonable, cutoff to think about the rare and common variants. But these days we have more samples and more samples. Like, now we have 1,000 samples, we have 10,000 samples, we have million samples. So people can go deeper and deeper. So now you define 1% for the threshold to distinguish common variants and rare variants. And more recently, people using the 0.1% for the rare variant and common variants. So you have to keep in mind that if you read some studies, and the study is actually using quite varying definition for the cutoff for rare variants and common variants, right? So, and this is the one way why about that situation. And if you look at the number of SNVs per individual, most of them are actually the common variants, like 3.3 million SNVs are actually common variants, right? And also these numbers are also quite varying by studies because we have the varying cutoff for the studies. But you can, instinctively think that the common variants probably the most of the variants constitute in the individuals, right? That is the one thing you can expect that. Okay, so selection and allele frequency, right? So allele frequency can be changed by selection. So what that means that if the allele is beneficial in the population, then their frequency will be increased over the time. In other words, if the alleles are deleterious in the traits, then the alleles probably removed over the time in a population, then the frequency is getting smaller and smaller, right? So, and that is the same meaning. And we discussed this part with the examples of achondroplasia and Marfan syndromes, and also in the sense of the polygenic model. And if you look at the natural selection, one key terminology you can think about is the fecundity, right? So fecundity is a measure of the fitness of trait, right? So fecundity is based on the fertility ratio. So fecundity is simply we can replace with the term fertility, right? And this is basically calculating the number of children in the group and compare with the general population. And that is the given the numbers of the birth in every year. So what that means, so if you look at the individuals diagnosed for depression, right? So if you look at the depression patient, right? And you measure their fertility. So how many depression patients have children in a year, and you then compare with the numbers from the general population, right? So general population is including some of the depressive people or other non-depression people, all mixed up there, just the general population. So if you look at here, the individuals diagnosed with depression, they have about maybe 10% or 20% reduction of the fertility compared to general population. But then if you look at the individuals diagnosed for schizophrenia or diagnosed for autism, then their fecundity is like a 50% or 75% of the decrease in that population. So what that means that the females diagnosed for schizophrenia or autism, they have the fecundity about 60% reduction, right? Or the males with the schizophrenia or autism, they have about 80% reduction of their fecundity. Why that happen? Schizophrenia or autism, their onset is way before the mating or way before the fertility period. What that means, when schizophrenia starts around teenagers, right? 70s or 18s, right? So that's a bit before the adulthood, right? And autism starts at 2 or 3 years old. It's normally diagnosed at the time. In Korean age, it's a 2, and is Western age, it's a 3 because we count differently. And then the schizophrenia or autism patients, they're developing their phenotype way before the mating period, around 20 or 30, right? So they have less chance to have mating, or they have the less chance to have the child because of the decreased probability of having mate, right? So why we measure the fecundity in a genetic studies, the reason is that whether that trait is undergo higher selective pressure or not, right? So that case, we measure the fecundity, and then we can think about how the genetics is working in the trait, right? So schizophrenia, the reduced fecundity is one of the hot topics in 1960s and 70s. Like, evolutionists like Julian Huxley, they raised the question, we see the reduced fecundity of the schizophrenia in our population, but why the people still keep having schizophrenia over the generation? How that can happen, right? Because in the sense of the natural selection, schizophrenia has to be selected out because of the natural selection mechanisms, right? But then schizophrenia keep appeared in our population. And what is the mechanism having the schizophrenia, the frequency maintained in our population, and why? That was the one question people have at the time. And autism didn't research very well in 1960s or 70s because, autism was a bit late constructed form. But around 90s or 2000, people studying cohort and people looking at their fecundity, and they report the reduced fecundity generally happen in neuropsychiatric disorder having, the early onsets like an autism, schizophrenia, intellectual disabilities. But why that happens in our population? And then people thought about another alternative hypothesis to explain the autism and schizophrenia, and that was the Nobel mutation we launched in shortly after. Right, so this is one of the landmark papers about the reduced fecundity of psychiatric disorder, published in 2013, the JAMA Psychiatry and by the poet. And this is the one of the great paper, but also the continuous report about the fecundity over the 20 years about this topic, right? So how we think about the fecundity and also the fitness, basically the similar meaning in allele frequency and genetic mutation, right? So another key terminology we can use in this situation is the penetrance, right? So penetrance in genetics is the proportion of the individual carrying a particular variant of a gene that also expressed an associated trait. So this is the tricky sentence. So the short example is the Fragile X. So Fragile X is the disease caused by single gene. and that is the single mutation too. So mutation in a FXS gene, and that appeared in early days, and then people has the Fragile X, and they died earlier, right? So they die earlier means that they have quite high reduced fecundity, and there's almost no chance to transmit their genotype to the next generation. So in other words, we can explain that they have higher selective pressure because natural selection is working in a Fragile X, right? And also the high fitness too. Or we can say that their fitness is quite low, right? So it's red colors on here, right? And another example is the Marfan syndrome. So Marfan syndrome, all the Marfan syndrome patients not died before the mating period. Some of them are they successful at the survival, but some of the 20%, 30% die before the 20 years old. So they have about moderate fitness, right? But this case, Marfan syndrome also has the mutation in one gene, right? And that gene can cause the disease, and that means that the mutation has to do something, right? So that means that the mutation in the Marfan syndrome also has high penetrance. So in other words, that is the same situation in Fragile X too, right? If the mutation, we know that causing the Fragile X or Marfan syndrome, they are actually doing something in that individual, right? They are determining that phenotype. So in that case, we say that the penetrance is almost 100%, right? But then we have the different opposition, right? So that is the Schizophrenia Mhc mutation. So that means that this is the mutation occurring in the MHC region, which is on the chromosome 6, right? So MHC is the genomic location for creating some genes for the immune system, but also this MHC region has lots of the mutation associated with schizophrenia. But this case, the mutation in the MHC, their penetrance is around 1 or 2%, right? The reason is that this MHC mutation is additive mutation. This mutation is working as polygenic situation. So that's why one single mutation is unlikely to determine disease, right? They are contributing to disease, but they are contributing en masse. They are contributing as a sum rather than a single particle, right? So this is the different situation as Marfan or Fragile X, because if the mutation wants to develop the schizophrenia risk, then the mutations has to be in a multiple, like a millions, and then they can develop the schizophrenia. So you can say that the moderate fitness can be observed in the schizophrenia, but this case, the mutation, a single mutation from this area has quite lower penetrance, right? So in other words, those mutations can be segregated to the next generation. So that means that it has the higher allele frequency compared to Fragile X mutation or Marfan syndrome mutation. So mutation seen in the Fragile X or Marfan syndrome, the allele frequency is almost zero, right? So it has only 1 or 2 individuals can be seen in large scale studies, right? So that case is different. And also we have wonderful examples for the counterpart, which is the Alzheimer, right? So Alzheimer, this is another well-known well-known disease, right? So when Alzheimer starts, after 60 years old or 65 years old. It's happened at quite late ages. So we called it late onset disease, right? But of course, Alzheimer can be, happen at 20 or 30 years old, but it's quite rare form. Most of the Alzheimer's is actually late onset Alzheimer, which is occurring at the 60 or 70, right? So in other words, Alzheimer is unlikely influencing mating situation, right? So like, Fragile X or autism or intellectual disabilities, it happens before the 10 years old, 15 years old. So the phenotypes is already fully developed in an individual. So it's unlikely have the proper mating in a society. But Alzheimer's is different, right? So if you think about the situation, like when you date with certain individual, and you normally talk about Netflix or the cool things in these days, right? Other than the question like whether your family has the Alzheimer or not, right? So if your boyfriend say my parents has the Alzheimer, then you're not gonna say break up, right? So this is the situation. So this is a likely impact on your transmission situation. So because it's the late onset disease, right? So normally that's why the late onset disease, or in other words, we say neurodegenerative disorders. Degenerative disorders means that after fully developed certain individuals, then we have the regression of the phenotype. So that's normally happen around 40s or 50s, right? So degeneration means that the late onset disease. That case, we're not gonna say this disease has the high selective pressure in their trait, right? Or another example is that the not that late onset, but it's quite late because it's developed in the teenagers. I don't know, around the teenagers, but some kids has too. And also quite high reproductive fitness. That means that it's not going to determine your mating situation. That is called the Archer syndrome, right? So this is like a K-pop. So Archer syndrome is that when you're exposed to sunshine, then you sneeze, right? So if you see the sun, or if you go outside, then if your skin is exposed to sun, then you sneeze, then we call it Archer, right? So this is like K-pop. And of course, then Archer syndrome is not going to determine your mating situation, right? So if you date with someone and you do sneeze over the sun, then your girlfriend is not gonna say, we have to break up, right? So this is the different, right? So Archer syndrome is also high reproductive fitness, and also their allele associated with Archer syndrome is highly transmit to the next generation. So there is no way we are impacting by that situation. Okay. Oh, why that happened here? So I don't know who did this. Okay, I can fix this. Okay, so next one is the genetic variant by transmission. So we are learning quite key concepts about the inherited variant and the novel variant, right? So another Winji for the Korean translation. So if you translate the genetic variants in Korean word, we call it 유전 변이. And this Korean translation is also bloody wrong. The reason is that the genetic variants, some of the genetic variants are not transmitted, but the word we use in Korean literally meaning transmission. So I don't know where the wrong things happen at the first time, but if you look at the genetic variants, not all of the genetic variants is transmitted to the next generation, right? And also all of the not genetic variants are inherited from the parents, right? So here we see the examples. Okay, so we have about 3 types of the transmission for genetic variant. So first one is the easiest one, which is the inherited variant. So inherited variant is the variant coming from one of the parents, right? So if you have father has the mutation coming to you, right? Or other types of the variants, and also well-known examples in our world is the de novo variant, right? Or we say de novo germline variants. So this is the variants sourced from the germ cells in your parents, right? So your mother or your father has the new mutation in their germ cells, and one of the germ cells accidentally put it into fertilization, then the mutation is coming to you, right? So their case is what we call the de novo variant. And other types of the de novo variant, which is the somatic variant, and or we say de novo somatic variant, right? So but people normally propel the short things, right? Somatic variant. So somatic variance is happening at the somatic cells over the lifetime, right? So this mutation is not from your parents, but it's accidentally happened in your, or stochastically happen in your cell, right? In your somatic cell, right? And this somatic variance is not subject to transmission to your offspring, right? So this is the different to the de novo variant. Okay, so first thing we have to remember that is that how the mutation is actually happening at the first time, right? So how the mutation happened? So the reason is that our DNA replication is not perfect process, right? So during the replication, we can have an error. So error means that the base pair mismatch between the strands, and the mismatch is the mutation in the form, right? So this error-prone process is the natural thing. This is the, always can happen during the replication. So what we have, so what biological organisms have in their system is that we have the proofreading system in our body, right? So proofreading means that if you do the DNA replication, then your body, your cells, your genome knows that error must be in your replicated DNA strand. So proofreading system going into that area and looking at the errors and fixing that mismatches. And you probably learn these systems in the molecular biology, right? But this proofreading system and fixing system is not always working in properly, right? It's also not the perfect situation. So in every DNA replication, we have a chance to have mutation, right? So this mutation, we call the new mutation in the cell division. So new means that in Latin, we call it de novo, right? So that's why we call the de novo mutation, right? And also these errors are different nucleotide changes, right? So sometimes it's A, sometimes it can happen T, G, or C. Sometimes it can happen as a indel form. Sometimes it can happen structural variance form, right? All of different formats we can have the mutation, right? And also the mutation rates are describing the situation, how many mutations actually occur during this replication process, right? So how many errors happen during this replication process, right? So if the mutation rate is very high, then that individuals, that organisms could be dead, right? Because too many mutations, right? The replication is not perfect, so they probably have very varying phenotype, and they are not have very stable situation for their development. So that case, the individual, the organism probably died, right? But so that's why if you look at the different species, they have different mutation rates, right? So we have different mutation rates to rabbits, mouse, or dogs, and they have all maintained their own ways of the mutation rate, right? Okay, so if that mutation, if that errors occur in somatic cells, we call it de novo somatic variant, right? And as I mentioned, that this is not heritable because it happened in your body cells, right? So this is not gonna be on your sperm in a transmission situation, right? But if the errors, these DNA replication errors occur in germ cells, in your sperm or in your eggs, then this could be de novo germline variants, and that is the heritable, right? It can be heritable if the sperm or eggs is conceived in the fertilization. Okay, so let's look at the somatic variants first. So we have lots of body cells in our body, right? So and I mean, as of that many body cells, we have the lots of chance to have many somatic, lots of the chance having the somatic mutation during lifetime, right? And this is the natural process, and we have certain, the rates of the mutation in our somatic cells, and we are totally tolerate with that situation. The reason is if the cells has the very bad mutation in one of our cells, then our cell system has really amazing defending mechanism for that situation, which is called the apoptosis, right? So if the cells totally change their properties by the mutation, then apoptosis is working, then totally kill that, the cells properly. Or autoimmune system can remove the cells from your body, right? But sometimes the somatic cells has totally crazy mutation and they are out of control, and these cells growing quickly and they become tumor, they become cancer, right? And some situation, the somatic mutation has to happen in a natural process or stochastically, but sometimes you are exposed to the certain, environmental exposures, right? Like, radiation or UV light or certain, whatever bad companies, the compounds to your body, then that could be mutagen, right? And this mutagen is totally out of the natural things, and that is changing your DNA in a crazy way. Then it can cause the cancers, right? So this is the way we are learning that the molecular biology stuffs and immune stuffs and related to these somatic mutations. And I'm probably sure you're covering these stuffs a lot in the subject. But one way we can note that is this mutagen or the somatic mutation the process in cancers has their own unique mutation signatures, right? So we call it mutation signatures, right? And they have lots of the CT conversion in a cancer cells. So if you look at the somatic mutation profile from certain cells, then you can expect that, whether these cells going to be in a cancer cells in a later stage or later developmental stage or something. So this is all, long been studies in the field, right? Then another types is the de novo mutation, right? So de novo mutation is, as I mentioned, that this is from the germ cells, and germ cells is obviously they have lots of cell division over the time, especially like sperms, right? Sperm has their own cell division every 16 days, right? So in an year there are 23 divisions and producing the new sperms in your body, right? And during that process, you have more chance of having mutations in these cell divisions, right? So if you have baby in a 20 years old, like males, they have in a single sperm, you have about 20 de novo mutations. So probably most of the students in the class, right? But about age 40s, the male sperm, like me, we have about like a 40 de novo mutation. The reason is that over the 20 years, we have more and more cell division in a sperm, and they have the accumulate of the de novo mutation over the time, right? Because we have long been processed, and there are long ways of the accumulation of the de novo mutation, and the proper reading system is not working properly over the time, right? So this is the way we have more de novo mutation in the later stages as well. But eggs are quite different because the eggs are having, halfway of the meiosis before the birth, right, in the womb, and they only have the rest half of their the germ cell division. So they have less chance to have that de novo mutation in their body, right? So how many de novo variants have in our genome? So we have about 70 de novo variants in our genome. So if you look at your genome and compare your parents, you have about 70 mutations totally different from your parents, right? So I have probably 70, you have probably 70, but your parents, let's say if your parents like, when they, have you at, 50 or 60, then you probably have more de novo variants than others, right? So when you say the mutation rates in our in our germ cell, we have about 0 to 1.0 to 1.5 times 10 to minus 8 per base pair per generation. So this is times by the human genome size, which is the 6 billion, right? 6 billion base pairs, right? And this 70 de novo variants is quite, consistent across the study. So I see these rates in my science paper, but also this is another my paper too, and other studies too. So we have about that rate. And interestingly, 80% of the de novo variants in you or in me or in other students is actually from fathers, because father has the long, way of having the de novo mutation accumulation in their sperm, right? And people had that question in earlier times, and when the genomic sequence technology released in the field, they studied that patterns using the large-scale population. So this is the data set from the Iceland, the Decode project, and they're looking at different parents, right? And they're looking at different families too, and they're looking at when they have the babies in their in their time, right? So this is the parents having the kids at the the mother is 20 and father is 20, and they have the mutation run 40 from father and probably 5 or 10 from mother, right? So this is about 50. But if you have the baby around 50 years old, but not data from female, but males, it has around 90 variants from the fathers, right? So if the father's age increase a year, then the mutation increase 1.5 every year. Mother is about 0.37. So we can say that there is a parental age effect in the de novo mutation, and this de novo mutation is doing something in our traits. And this is one of the social phenomena we are facing in these days, because the reason is, if you look at the news media, our, the first marriage is getting late and late. And this, when I was in 20s, people worried about that you guys marry around, 29 or 30. But nowadays it's like 34, 35, right? So the way they quite delay their parental age is, I think, is one of these serious issues in terms of the genetic disease, right? Okay, so that was the same thing. Okay, so de novo mutation. So de novo mutation is quite small numbers, and given the 3 million or 4 million mutations in our genome, de novo mutation only has 70 mutations in your genome, right? But this 70 mutation does not subject to the natural selection, right? So this mutation doing something in your phenotypic development, there is no way we can think about the natural selection, right? So this is fully developed that phenotype in your body, right? So over the 10 years, people study a lot about de novo mutation, including me, right? And what we found that the de novo mutation is a main cause of the severe phenotype in development, like Down syndrome. Down syndrome is the trisomy in chromosome 21, and there is also one of the de novo chromosomal aberration, right? This is earlier described, right? And also autism, also the main reason is the de novo mutation. Epilepsy too, congenital heart disease, also de novo mutation too. Lots of the congenital situations we can describe by de novo mutation. The reason why is that de novo mutation was not selected to natural selection, and there is, generate a phenotype, directly when you have the mutation in a critical gene, right? So de novo mutation is the one way we are probably keep mentioning in our field. Okay, so as I mentioned, that the rare risk variants like the de novo mutation have the great potential to carry risk for disorders. So we are talking about the 72 novel mutation, right? That is the almost the zero chance to have the same mutation in our population because this is totally new mutation and unique mutation, right? And also you have to link the idea from this 72 novel mutation to mutational model, right? So you probably think about the Kimura-Muto's neutral mutation model, right? So given that model, we have the 72 novel mutation, but not all of these 70 mutations doing something in our phenotypes, right? In our traits. The reason is that most of the mutation is actually neutral, including the de novo mutation. So if you look at the patients with the autism or epilepsy, they don't have the 70 deleterious de novo mutation. They have one de novo mutation or 0 de novo mutation, right? So the way de novo mutation occurring some disease, they only contribute 1 or 0 to the population, right? So this is also linking idea to mutation model. Okay, so and the rare variants and common variants, one individual, right? Or you can also put the graph like this for the allele frequency and the risk of the variants. In other words, we call the effect size, or in other words, we call the penetrance. So they are quite, equivalent, terminology describing the same things, right? So this is another landmark papers. I mean, if you are seriously interested in the biomedical science, not just genetics, if you are interested in disease studies, or if you are interested in clinical stuff, I think this is the must-read paper. This is the hesitation and doubts on the genetic studies in early days by many humans, and the reason is that I pointed out this paper is one of the sort of the transition or the cornerstones of the biomedical science. The reason is that this is the period when the next generation sequencing technology, so new genomic technology coming out to the field, and people thought that if you use that technology, we can all conquer, we can all find the rare Mendelian variants to the Mendelian disease, and we conquer that disease, and we develop the drug, we develop the trait, and we overcome everything, right? And in other parts, people doing the additive genetics, and in other words, we say common variants. We're going to cover this in the later section. But anyway, so this period, people say that things not gonna working in that way. So you have to think about rare variants, but also the common variants. So some people study in this perspective, some people study in this perspective, and this paper, people has their conflicting view on the genetics and biomedical science, and given this paper, people keep debating which one is the best way we can think about disease in the clinical studies, right? The short spoiler for that, there's no correct answer from these things, but this is the conceptually summarized that the variant frequency and the penetrance for the disease has the inverse relationship. That is true, but what's not true is that in a same disease, in the same disease, we have certain portion of the rare variant allele, we have the certain portion of the common variant allele, which we see in the examples of height, right? So we're going to cover that issue in the later section, but if you are burning desire in biomedical science, not just the genetics, but also the field, this is the things you have to keep in your mind, because this is one of the way we did the sort of the big transition in a biomedical science. So please have the review for yourself. Okay, so we are continuing on the genetic variants by consequence, right? So we can also summarize the relationship of the variants by their consequence in a protein sequence, right? So as you know that there is a central dogma and there is a information flows from the DNA transcription to translation, right? And we also look at the examples of how these variants can occur the phenotype. So we looked at the relationship between the genotype and phenotype, right? Okay, so one way we can think about is the protein made of the amino acid sequence based on the triplet codon, which is the 3 nucleotides of the DNA, and it encoding the one amino acid. So if the CGG in the DNA and that producing the arginine, but if that is the G changed to A, then it produced the glutamine, right? So one variant change can happen in a protein sequence, and it can determine the phenotype as an allele, right? So we learned about the some examples of how mutation can be work as an allele in a modern sense, and here is one example from the genetic testing. So you have the genetic testing, and you have the mutation in this position, and that is G to A changes, right? And how that is working, and it actually working in the 2558 position of your cDNA, which is transcribed from the mRNA, right? And that is also changed G to A, right? And that is in the protein, it changed the one amino acid on 8053 position, and it changed arginine to glutamine, right? So your mutation can change the phenotype ultimately. So this is the way how we think about the consequence. So consequence means the result of the mutation, right? So we can think about the consequence of the mutation from the from each situation. And some of the mutations also working as a producing the stop codon. So that's also given the triplet codon information, and also we are learning the situation for the developmental disorder situation. So if you look at the mutation, you can actually describe many ways to think about that mutation consequence, right? So here you have the gene, and here you have the transcript of one gene, right? So if the mutation are happening in exonic position, which is in exon, right? You learned about exon, right? And that is, you called it coding mutation, like missense mutation or stop gain mutation, frame shift mutation, or all sorts of the describing the coding exonic triplet codon situations, right? But there are different compartments in the transcript, like introns, right? Intron is the position between exons, right? So between exons, if the mutation happen in the intron, we call the intron variant, right? But sometimes the mutation can happen on the boundary between the exon and intron. So that case, this boundary we call the splicing sites, right? You learned from the molecular biology. So splicing sites means that generating the different splicing isoforms from the transcription situation. So that case, you call the splice donor, the first position, 5', and the 3' is the splicing acceptors, right? So that's why they join the splicing format. So that case, we can specifically call this splicing donor variants, right? Or if you look at the transcript, there are some regions called untranslated regions. So on a 5' end, if the mutation happened there, that's what we call the 5' UTR variants or 3' UTR variants, right? So it depends on which end of your UTRs. Or mutation can happen outside the transcript, right? So outside the transcript, we call the introgenic region. The reason is that if you have the gene here, if you have the gene, another position here, then between the gene, so between the gene means the introgenic. So inter means between, the genic means that two genes, right? So that case we call the introgenic variants. But interesting stuff is that if, as you learned from the molecular biology, you have the transcription start sites just before the first position of your transcript, right? Because this position is the place where RNA polymerase II binds to that place and initiate the transcript, right? So that case, you can specifically call this area is the promoters, right? So promoters is quite varying definition. So where they located in the transcription site. So we bluntly call that positions as a upstream gene region. So upstream means the upstream of the transcript, right? So if the mutation happened in that area, then we call it upstream gene variant, right? Or if the mutation happened after the transcript, then we call it downstream gene variant, right? So we have some mutation happen in the coding region, but also outside the coding region. So outside the exonic region. So that case we call the non-coding variant, right? So why we care about the non-coding variants? Because the non-coding variants have lots of the regulatory elements. As I mentioned, they have the promoters, they have the transcription factor binding site, which is when RNA polymerase II binds to that area, there are accessory proteins binds to the area too, alongside with the RNA polymerase II. So that case, we call the transcription factor binding sites because those proteins helping the transcription situation. And also we have some of the regulatory sequence like an enhancers, right? So enhancing the transcription. So that case, we also collectively call it enhancers, right? So all of these regulatory elements can located in the non-coding region. And if the mutation happened in this non-coding region, then we care about their consequence, and that mutation can occur in the phenotypic changes as well. So I don't think I put the hyperlink here. I'll put the hyperlink so you can read about what is the differences between this terminology. And there are lots of terminology, but once you understand the concept of them, according to the transcript structure, then you probably understand what each of them meaning in each situation, because this is literally indicating the position of the mutation. So I'll update the link for this. Okay, so why we care about the variant consequence? The reason is that some of the variants is critical to the phenotypic development or the trait variants, but some of the variants is not, that critical. They could be quite marginal in a trait, right? So this is the study from the Iceland again, and they analyzed the 2,000 Icelandic people, and they're looking at the around 100 quantitative and binary traits from their health record, right? And they measure how much each variant consequence changing the traits in the health information, right? So someone simply can ask you, which types of the variants is really critical to trait development, right? So we can intuitively answer that probably protein truncate mutation, which is the loss of function mutation, or stop gain mutation, which is the introducing the stop codon in a coding sequence, right? That case, they are changing the health record a lot. And if you look at the data set, indeed, it's yes, right? So loss of function mutation, they actually change lots of the health traits, right? And the missense mutation, which is one change of amino acid, they did some. And splice region also changing some protein sequence as well. It's yes, right? But other non-coding mutation or synonymous mutation, they don't do lots of things, right? So they getting less impact on the trait values, right? Okay, so one way we can think about the coding variants are likely to impact more on the traits because they produce the protein, and this protein is the direct contributor to the phenotype, right? So this is the obvious way we can think about this mutation is impacting on the trait values, right? So this could be one reason for this why. Okay, and another important things you have to keep in mind that the variant consequence is it can be differed by transcript. right? Because if you look at the same gene, so this is the gene A, so they are the same gene, but the same gene can have multiple different isoforms, right? Isoform 1, 2, and 3 because of the splicing, alternative splicing process, right? So this isoform has 5 exons. Isoform 2 has 4 exons, don't have the second exon, and isoform 3 has only 3 exons, skipping first and second exons, right? So if the mutation happened in here, and this mutation can be coding mutation according to isoform 1, but according to isoform 2, this mutation is intronic mutation. And according to isoform 3, this mutation can be upstream mutation, right? So how can you judge, which variant consequence this mutation have, right? And also this is the much complicated consideration if you want to apply in the clinical situation. The reason is that if the isoform 1 and 2 is not, okay, so let's think about this situation. So if you find this mutation from the patients with the neurological disorders, right? And you want to find the cause of the, the disease, right? And this is the neurological condition. So you want to look at the brain expressed gene, right? So if you look at these 3 isoforms, isoform 1 and 2 is not expressed in brain, and isoform 3 is expressed in brain. So in that case, you have to estimate the variant consequence based on this isoform 3, right? But in reality, it's not always working that way. So there is a different ways of doing algorithms in estimating the variant consequence. So there are, the ways used to improving the algorithm to estimate the variant annotation in a clinical situation. So that's why I introduced this skip for your information. Okay, so we covered the 4 scenarios of looking at the variant classification, and there are some, the great books and textbooks, and also the reviews I explained in here. So if you can have the research by yourself, that will be great. And also, if you have the questions, then it's always welcome to leave yours on the Slack channel. Then I can follow up afterwards. Okay, so I think it's 1 minute past. Okay, so see you on Wednesday.